Welcome to this explainer. Today we're profiling some of the absolute most successful thieves on the entire planet. But, we're not talking about bank robbers here. No, we are diving into a fascinating biological true crime profile of microscopic infiltrators that actually target the human body. We're talking about small intestinal nematodes. These organisms execute a perfectly evolved high-stakes heist right inside our digestive tracts, basically stealing our nutrients and completely evading our defenses.
Okay, let's dive into the clinical dossier for today's heist. First up, we'll meet the intestinal nematodes themselves. Then, we'll take a really close look at the Ascaris roundworm, followed by hookworms and Strongyloides. After that, we'll cover diagnosing the infection, and finally wrap up with strategies for prevention and control.
All right, Section 1. Meet the intestinal nematodes, the geohelminths.
So, let's identify the main suspects in our biological heist. Ascaris lumbricoides, hookworms like Nematodes americanus, and Strongyloides stricoralis. To really wrap our heads around these parasites, we have to ground ourselves in the environmental aspect of their threat. These suspects belong to a medically important group known as geohelminths, and that prefix geo is totally key here. It means their life cycle has a strictly non-negotiable requirement. They absolutely must have a development phase in the soil before they can even infect a human. They literally use the very earth we walk on and the dirt we touch as their main staging ground for the invasion.
Moving on to Section 2, Ascaris, the giant roundworm.
Right, so this first infiltrator is incredibly persistent. An adult female Ascaris produces a massive amount of eggs every single day. And those fertilized eggs, they're so resilient they can remain totally viable in dust and soil for several years, just waiting for a host. The infiltration route is frankly bizarre. It kicks off when embryonated eggs in contaminated soil are ingested orally. But get this, they don't just stay in the gut. Once inside, the larvae hatch and go on this wild migration straight through the lungs. They actively rupture your capillaries to enter the alveolar spaces, causing microhemorrhage in the lung tissue. And while they're hanging out in the lungs, they actually undergo two moltings, releasing a highly allergic molting fluid. This fluid triggers pulmonary symptoms. We're talking severe coughs, wheezing, and a condition called eosinophilic pulmonitis, which is basically an intense accumulation of white blood cells in the lungs. Finally, they travel all the way up the windpipe just to be swallowed again, settling down permanently in the small intestine.
Mapping out this exact journey is pretty wild. You start down at the embryonic egg, the infective stage, being ingested. It hits the bloodstream, takes that detour to the lungs for a highly disruptive maturation phase, gets coughed up, swallowed right back down the pharynx, and finally ends up in the small intestine as a full-grown adult. Then, it drops eggs back into the external environment via feces, completely closing the loop.
Once they're established in the small intestine, adult Ascaris worms can live for 1 to 2 years, absorbing nutrition directly from our digested and semi-digested food. In heavy infections, they literally cover the entire intestinal absorption area. Now, this nutritional theft has tragic consequences, especially for children. It causes severe nutritional deficiencies, leading to stunted growth and cognitive retardation in preschool and early school kids. And if there are enough worms present, they can physically tangle together and cause a total intestinal obstruction. The reality of that kind of obstruction is visually shocking. Severe Ascaris infections can literally require surgical extraction directly from the jejunum of the small intestine. And because these worms are highly mobile, they don't always stay put. Migrating adults can actually block the biliary tract, causing cholangitis, which is an inflammation of the bile duct system. They can trigger pancreatitis, a severe inflammation of the pancreas, or in extreme cases, they can even be expelled orally straight through the mouth. It is a massive physical and nutritional burden on the host.
Let's shift gears to Section 3, hookworms and Strongyloides.
Okay, so now we're looking at our next suspects, hookworms, specifically Nematodes americanus and Ancylostoma duodenale, and Strongyloides. Now, hookworms operate with a radically different mode of entry. While Ascaris infiltrates because you swallow contaminated soil, hookworms use what's called percutaneous entry. This means their infective stage, the filiform larva, actively burrows directly through thin, intact areas of human skin. This usually happens right on the foot, specifically the top of the foot or the toe web, when someone walks barefoot on infected soil. The hands can also be an entry point. The physical burrowing of these larvae causes localized itching at the entry point, a symptom clinically known in the field as ground itch or dermatitis. To identify this specific thief, medical detectives look for its microscopic signature, the hookworm ovum. It has a very distinct oval shape. measuring about 65 by 40 micrometers. It's totally colorless with a very thin shell that looks like a distinct black line right around the edge. Inside, the ovum appears segmented. And what's absolutely crucial for medical labs to note is that in older feces samples, the larva may have actually already developed inside the egg.
Then we have Strongyloides stercoralis. We can basically call this one the master of autoinfection. Its life cycle is incredibly complex, alternating between free-living and parasitic cycles, but the absolute most crucial point here is its ability to auto-infect. Inside the host's intestine, first stage rhabditiform larvae, which is the early, non-infective stage, can develop directly into infective filiform larvae right there in the gut. This biological loophole is a real game changer. It allows untreated infections to persist for many, many years, constantly reinfecting the host without them ever needing to touch external soil again.
Moving right along to Section 4, diagnosing the infection.
So, how do we actually catch them? Medical detectives rely primarily on identifying the microscopic clues these thieves leave behind in the stool, typically by using a direct saline smear. Let's look at the specific clues left by Ascaris. Medical professionals have to differentiate between two distinct types of eggs under the microscope. A fertilized egg is yellow-brown, oval, or round, and covered by a very uneven, thickened coat known as a mammillated shell. Inside, it contains an unsegmented ovum. Conversely, the unfertilized egg is far more elongated. It's often described as barrel-shaped, with a noticeably thinner wall and a much more granular coat.
The visual contrast between the two is stark. With the fertilized egg, which measures about 50 to 70 micrometers long, you clearly see that thick, bumpy, uneven mammillated shell. But the unfertilized egg looks noticeably stretched out, barrel-shaped, like we said, with that thinner wall and large central granules. Recognizing these highly distinct visual signatures is exactly how a lab confirms the exact nature of the heist.
But diagnosing these cases comes with some pretty high-stakes lab protocols. Remember how we said adult worms are sometimes expelled through the anus or mouth? Well, they resemble pinkish earthworms, tapering at both ends, and they can be huge, measuring 12 to 35 centimeters. If they're recovered, lab techs absolutely must handle them with forceps, because direct skin contact can cause severe allergic reactions. Furthermore, if a lab is storing eggs in formal saline, they have to treat the fecal deposit with hot formal saline, specifically between 70 and 80 degrees Celsius. Why? Because this extreme heat prevents the eggs from developing into their highly infective stage right there in the laboratory.
And that brings us to Section 5, prevention and control.
Once we truly understand how these geohelminths operate, we can effectively lock these infiltrators out and protect vulnerable populations. Locking out the invaders relies entirely on breaking their life cycle, which, as we established earlier, centers completely around the soil. So the primary defense is preventing the soil from becoming fecally polluted in the first place by improving environmental sanitation and using proper toilets. Personal hygiene is just as critical here, washing your hands before eating and strictly avoiding uncooked vegetables, green salads, and fruits that might be contaminated with egg-laden feces. And remember those hookworms that enter percutaneously? We block them simply by wearing adequate protective footwear to stop those larvae from ever penetrating the feet. Finally, actively treating infected individuals and providing widespread health education cuts off the source of new eggs.
These microscopic thieves have evolved brilliant, complex, and honestly devastating ways to infiltrate our bodies, from burrowing straight through our toes to migrating all the way through our lungs. But their greatest weakness is simple sanitation. So, are your daily habits and environment leaving the door open for these microscopic infiltrators? By understanding their methods, we realize that basic hygiene, clean water, and proper footwear aren't just modern conveniences. They are quite literally our frontline biological defenses. Thank you so much for joining me on this explainer. Keep learning and stay curious.